So get this. The other day I'm in my kitchen, right? It's early. I haven't had my coffee. I'm trying to make some toast. I put the bread in. I push the little lever down. Nothing happens. I jiggle it. I unplug it. I plug it back in. Still nothing. Then I hear a tiny voice. The myelin sheath is compromised. I look around. Nobody's there. I look at my dog, Gary. He looks back like, dude, are you gonna share that bacon or what? Okay, maybe I need more sleep. But then, again, from the toaster, we must reprogram the T cells. That's when I realized my toaster thinks it's a geneticist trying to cure multiple sclerosis. Now, I know what you're thinking. Jimmy, that's insane. And you're right. But it got me thinking. What if we could just reprogram things when they go wrong in our bodies? What if your internal wiring gets frayed, like an MS, and you could send in a tiny repairman? Sounds like sci-fi, right? Like The Rock shrunk to a tic-tac fighting a rogue antibody. But here's the crazy part. It's not a movie. It's real. It's happening right now, probably in a lab that smells faintly of burnt coffee. And pure genius. This sent me down a rabbit hole. I started reading about multiple sclerosis. MS. Basically, your body's defense team, your immune system, gets confused. It starts thinking your myelin, the protective coating on your nerves, is the bad guy. So, it attacks. Alright, so what in the world is CRISPR? Let's break it down. Imagine your DNA is the world's longest, most complicated IKEA instruction manual. It has billions of steps to build and run you. Now imagine there's a typo on page 4,782,951. Instead of saying attach leg A to table B, it says attach leg A to your dog. Suddenly, your furniture is a mess, and your dog is very, very annoyed. That typo is like a faulty gene that can cause a disease. For years, scientists could see the typo, but they couldn't fix it. They could only try to deal with the angry dog. Enter CRISPR. CRISPR is like a microscopic superhero duo. The first part is a guide. Let's call him Finder Frank. Frank is like a super nerd with a GPS. You give him a picture of the typo, that bad gene, and he will scan the entire IKEA manual of your DNA until he finds the exact spot. He's relentless. He has one mission and one mission only. Find that specific, troublemaking sentence. He's the guy at the party who only wants to talk about one scene from Star Wars for 3 hours. He is very, very focused. Once Finder Frank locates the typo, he calls in his partner. This is the muscle of the operation, a pair of molecular scissors. Let's call her Snippy Sue. And with a satisfying snip, Sue cuts the DNA right where the typo is. The cell's natural repair system then jumps in and, most of the time, patches it up correctly. It's like you used whiteout on the IKEA manual and wrote in the right instruction. Suddenly, your dog is safe and your table has all its legs. And that's exactly what scientists are trying to do with MS. Okay, so we have our tiny superhero, CRISPR. Now, how do we use it to fight a big, complicated baddie like multiple sclerosis? Remember, with MS, the problem is your own immune system's T cells have gone rogue. They're like bouncers at a club who suddenly start tackling the club owner. They're supposed to be protecting the place, but they've got the wrong instructions. Their internal GPS is telling them that myelin, the cozy insulation around your nerves, is a dangerous intruder. The mission, should CRISPR choose to accept it, is to sneak into these confused T cells and fix their navigation system. The plan is brilliant and a little bit sneaky. First, scientists take a blood sample from a person with MS. They're not just taking any blood, they're hunting for those specific, confused T cells. It's like trying to find all the guys wearing the wrong uniform in a giant army. Once they've rounded up these troublemakers, they take them to the lab. This is where the magic happens. The lab becomes a sort of re-education camp for cells. Think of it like a timeout, but instead of sitting in a corner, the cells are about to get a major software update. This is where Finder Frank gets to work, and Snippy Sue gets to work. Inside the lab, scientists introduce the CRISPR system to the captured T cells. They program Finder Frank with the genetic address of the faulty receptor, the part of the T cell that's mistakenly identifying myelin as an enemy. Frank zips into the cell's DNA, finds that exact sequence, and plants a big flashing arrow on it. Then, Snippy Sue comes in and she cuts out the faulty gene. The cell, being the clever little thing it is, then repairs the cut. The big reveal! So, we've got the plan, we've got the science, and we've got the tiny, clumsy superhero. But does it actually work? Is this all just a fun story, or are we actually fixing things? Well, folks, I'm here to tell you, it's not just a story. We're seeing real success. The results are still early, but they are incredibly exciting. Scientists have been testing this out in labs using cells and animal models. Researchers took those confused, myelin-attacking T cells from mice that have a disease very similar to MS. They performed the CRISPR surgery, reprogramming the cells so they would stop their attack. Then, they put these newly edited peaceful cells back into the sick mice. And what happened next was amazing. The disease progression in the mice not only stopped, but in some cases, it actually reversed. The mice started to get better. Their symptoms improved. It was a huge breakthrough. Think about what that means. They didn't just treat the symptoms with a pill. They went to the source code of the disease and rewrote it. It's the difference between constantly patching a leaky roof and just building a brand new solid roof. One is a temporary fix, the other is a potential cure. This success in mice was a massive proof of concept. It's a practical strategy that could one day be used in humans. It's the first step on a very long journey, but it's a gigantic one. Of course, mice aren't people, but this research is the green light that scientists were waiting for. Now, it's easy to imagine scientists as these flawless geniuses in pristine labs. But let's be real, science is messy. It's full of trial and error. For every brilliant breakthrough, there are probably 100 experiments that ended with a weird smell and someone quietly muttering, "Well, let's not do that again." The researchers working on CRISPR for MS are facing some huge challenges. It's not as simple as just pointing Snippy Sue at a cell and letting her go wild. One of the biggest hurdles is making sure she cuts in the right place, and only the right place. This is what they call off-target effects. Imagine you tell your robot vacuum to clean up a Cheerio you dropped, but instead, it eats your Wi-Fi router. It tangles your shoelaces. It swallows the TV remote. That's an off-target effect. With CRISPR, there's a small chance Snippy Sue might snip a healthy part of your DNA. This could cause a whole new set of problems. So, scientists are working tirelessly to make the system more accurate. They're essentially upgrading Finder Frank's GPS from a flip phone map to a super advanced, satellite-guided system. It needs to distinguish between two nearly identical streets in the vast city of your genome. Another challenge is delivery. How do you get the CRISPR machinery to the right cells inside a living, breathing person? It's one thing to do it in a petri dish in a lab, but another to do it inside a body with 37 trillion cells. It's like trying to deliver a tiny pizza to one specific apartment in New York City, but you don't have the address, and the pizza box dissolves in the rain. Scientists are experimenting with different delivery vehicles, like harmless viruses that are hollowed out and used as tiny Trojan horses to carry the CRISPR system into the target T cells. It's a biological special delivery service. And finally, there's the question of permanence and safety. How long will the edit last? Will the reprogrammed cells stick around? And what are the long-term consequences of editing human DNA? These are massive ethical and scientific questions. So, there you have it. A secret war being waged inside our bodies. Tiny gene-editing superheroes fighting to fix a terrible mistake. It's a story of hope. It's a story of incredible ingenuity. From my crazy toaster to the labs where this is actually happening. The world of science is more amazing and funnier than you could ever imagine. And this is just one story. There are thousands more just like it, happening in every field of medicine. We're living in an age of miracles, people. I want to be your guide through all of it. If you had as much fun learning about Snippy Sue as I did, then you're in the right place. So, do me a favor, hit that subscribe button. Give this video a big thumbs up. Ring that notification bell like you're a game show contestant who just won the grand prize. It tells me you want more of this stuff. It helps other people find their way to this little corner of awesome, hilarious science. Seriously, you don't want to miss what's coming next. We're just scratching the surface here. The same CRISPR technology we talked about today isn't just for MS sickle cell anemia, Huntington's disease, some types of cancer. It's like they've discovered a Swiss Army knife for our DNA. Every week there's a new discovery, a new possibility. And for our next trick, what if I told you the next generation of CRISPR might not even need to cut the DNA at all? That's right. Scientists are working on something called base editing. It doesn't use Snippy Sue's scissors. Instead, it's like a tiny pencil with an eraser that can chemically rewrite a single letter of DNA without breaking the strand. It's like fixing a typo without leaving any whiteout marks. You'll have to stick around to find out. The future is coming, and it's going to be epic. See you next time.